The overall goal of this procedure is to identify and quantitate microRNAs present in exosomes obtained from blood or cell culture media. This is accomplished by first purifying exosomes from blood or cell culture media using differential centrifugation. The second step is to purify total exosomal RNA using a microRNA microRNA isolation kit. After confirming the integrity and concentration of the purified RNA, it is converted to cDNA using reagents in the megaplex reverse transcriptase reaction kit. Next, a preamplification step is performed using megaplex preamp primers and a TACMan preamp master mix to amplify the microRNAs. The final step is to run a real-time PCR reaction using two 384-well microfluidic cards or TACMan low-density array cards containing dried TACMan primer probes. Ultimately, this procedure will enable the detection of up to 758 known microRNAs.
This technique measures variations in exosomal microRNA content that are dependent on the source and physiological conditions under which they are secreted. The implications of quantifying these unique signatures extend toward development of biomarkers and novel therapeutic intervention strategies.
To begin this procedure, invert an EDTA-coated tube containing 10 milliliters of whole blood 5 times to ensure mixing of the anticoagulant agent and place it upright at room temperature for 10 to 60 minutes. Then, centrifuge the sample at 2000 times G and 4 degrees Celsius for 15 minutes to separate the blood. After 15 minutes, remove the tube from the centrifuge and collect the plasma in a new 15 milliliter tube. Place the plasma on ice and dilute it with an equal volume of PBS. Then, gently mix the sample and place it into the centrifuge. Centrifuge at 2000 times G and 4 degrees Celsius for 30 minutes. Next, remove the sample from the centrifuge. Transfer the supernatant to a new centrifuge tube, add 1x PBS to 24 milliliters, and centrifuge at 12,000 times G and 4 degrees Celsius for 45 minutes. Then, transfer the supernatant to an ultracentrifuge tube and centrifuge at 110,000 times G and 4 degrees Celsius for 2 hours. After 2 hours, remove the sample from the ultracentrifuge and remove the supernatant carefully with a pipette. Resuspend the pellet in chilled PBS and place the sample back into the ultracentrifuge. Spin at 100,000 times G and 4 degrees Celsius for 1 hour. Following centrifugation, remove the supernatant carefully with a pipette and resuspend the pellet in 300 microliters of RNA lysis buffer. Alternatively, the sample can be suspended in PBS or RIPA buffer for electron microscopy and Western blot analysis, respectively. Prepare the area for isolating RNA by wearing personal protective equipment, wiping surfaces and pipettes with RNase ZAP, and using sterile filter tips and tubes. Then, determine if preamplification of RNA is necessary by quantifying the isolated RNA with a spectrometer. For samples with greater than 350 nanograms of RNA, preamplification may not be necessary unless the transcript of interest is in low abundance. Next, prepare cDNA from purified RNA using Megaplex Pool's reverse transcriptase primers. Start by thawing the primers on ice. Pool A and Pool B contain primers for the microRNAs on microfluidic cards array A and B. While they thaw, label 2 RNase-free 1.5 milliliter tubes for the reverse transcription with Pool A primers or Pool B primers, and prepare master mix for each separately from the components as described in the text protocol. Be careful to keep the Pool A and Pool B primers separate. Then, invert the tubes with the master mix 6 times and centrifuge briefly. pipette 4.5 microliters of the RT reaction mix into each tube of the microamp 8-tube strips. Next, add 3 microliters of the RNA sample containing between 1 and 350 nanograms of RNA to each tube. Stir the sample with the pipette tip and cap the tubes. Spin the samples down briefly and incubate them on ice for 5 minutes. After the incubation, load the reaction tubes into the PCR machine and run it under the following conditions. When the PCR has completed, store the cDNA at -20 degrees Celsius for up to 1 week or continue to preamplification. For samples with less than 350 nanograms, or if the transcript of interest is in low abundance, perform the following preamplification steps. Begin by thawing the preamp primers for Pool A and Pool B on ice and invert them to mix. Next, swirl the TACMAN preamp master mix and keep it on ice also. Then, prepare the preamplification master mix. Next, pipette 2.5 microliters of the cDNA product into microamp 8-tube strips, and then add 22.5 microliters of the preamplification reaction master mix. Finally, incubate the tubes on ice for 5 minutes. Then, load the sample into the PCR machine and run it under the following conditions. After the preamplification reaction is finished, briefly centrifuge the tubes, and then add 75 microliters of 0.1x tris EDTA pH 8.0 to each tube. Next, mix the tubes briefly and spin them down. Continue on to load the TACMAN microRNA array or store the diluted product for up to 1 week at -20 degrees Celsius. To prepare the samples for the TACMAN microRNA array, combine 450 microliters of the TACMAN Universal PCR Master Mix, 9 microliters of the diluted preamplification product, and 441 microliters of nuclease-free water to an RNase-free 1.5 milliliter tube on ice. Then, mix the tube by inverting it 6 times and centrifuge the sample briefly. Dispense 100 microliters of the PCR reaction mix into each port of the TACMAN microRNA array card. Next, centrifuge the card twice at 1000 times G for 1 minute, and then seal the card. Next, load the array into the thermocycler. Next, run the samples using the default thermal cycling conditions provided with the primers in the Applied Biosystems manual for the 384-well TACMAN low-density array. In order to run mRNA analysis in the Applied Biosystems 7900 HG fast real-time PCR system, first change the heat blocks to run a 96-well plate. The transmission electron microscope image shown on the left is of exosomes isolated from mouse blood using the methods described in this video. The image on the right is of exosomes purified from cell culture media and then gold-labeled anti-CD81. Exosomes typically range from 30 to 100 nanometers in diameter. The relative abundance of normalized microRNAs found in exosomes isolated from rodent blood, and human aortic endothelial cell culture media are shown here as delta CT values normalized to U6 RNA endogenous control. Positive delta CT values indicate that the microRNA is less abundant, and negative delta CT values indicate that the microRNA is more abundant. Not all microRNAs are present in each sample type. For example, miR-126 was not detected in rodent blood exosomes, and miR-200C is absent from HAOEC exosomes. The remaining 4 microRNAs are present in all 3 samples in varying amounts. Although this protocol focuses on characterizing exosomal microRNAs, individual mRNA transcripts can also be quantified from exosomes using qPCR. When compared with mRNA from whole blood, exosomal RNA showed similar expression of both TNF-alpha and vascular endothelial growth factor A.
Combining these techniques has paved the way for researchers in the field of RNA biology to explore the function of circulating RNAs in mediating gene expression changes and in identifying molecular signatures associated with exosomes in both normal and diseased states.